It's going. So good morning. My name is Naveed Aziz, and I'm the Chief Administrative and Scientific Officer of Canada's Genomics Enterprise. I'd like to start by thanking Paul for giving me the opportunity to speak to all of you today. So in the next 15 or so minutes, hopefully 15, I'll tell you a little bit about who we are, what we do, and then also tell you a little bit about our initiative called CanSeq 150, which I hope will be relevant and of interest to this audience. So let's get going. So Canada Genomics Enterprise, or CGEn, is a national sequencing platform with nodes in Toronto, Montreal, and Vancouver. Our mission is to support Canadian genomic science by building and supporting a large national sequencing platform to enhance Canada's capacity in sequencing and analysis. To give you an idea, we have over 200 staff employed at the 3 CGEn sites. And to give you an idea of the scale of our operation, just some data from I pulled out for our last year's activities. So we service around 1,864 principal investigator labs. So these are separate labs that have come to us for service. We have trained over 200 people, and these are the people not that are involved as CGEn staff, but these are people who come to our labs for training, get to learn some techniques and processes, and then go back to their labs to practice them. Obviously, the data we generate is used by many people. I don't want to go into these numbers. Output-wise, so in the last year, we generated over 1,230 terabytes of data. So just to give you an idea, that will be equal to, did some calculations the other day, that will be equal to 500,000 HD movies on Netflix. 500,000. And if you sat down to watch them back to back to back to back without a break, it will take you over 85 years to finish them off.
Start early.
Yeah, so that's what we just generated in the last year. And we obviously work on a fee-for-service basis. We generated about $39 million just for fee-for-service. And then we published over 100 papers by our own staff on technologies and techniques. And there were 400 or so papers that cited us as being their partners. We think it's a huge underrepresentation because I have been an academic. We all know we forget to acknowledge people who need to be acknowledged. Technology-wise, spread across the country, as I said, Vancouver, Toronto, Montreal. We've got a whole host of genomic sequencing and other genomic technologies within the 3 sites, all type of sequencing technologies that are out there right now. If it's not up in at production scale, we have it as for testing. And output capacity-wise, we have a capacity of delivering over 40,000 human 30x equivalent genomes a year. And for the nerds in the room, we have over 50,000 hyper-threaded cores and over 26 petabytes of storage between the 3 sites. And our partners and users come from all walks of life. We work with academia, we work with industry and nonprofit organizations, provincial and federal organizations, international projects, consortia. We do a lot of work for the NIH, although they have their own sequencing facilities in the US, but just because of the expertise some of the people in our nodes offer, some of the NIH projects come to us as well. I think that's enough about who we are, and if you're interested more, I'm here at least till lunch and maybe a little bit after, so please let me know and we can talk about it. So CanSeq 150. So this is a project that we launched earlier this year in February with the idea to sequence 150 new genomes that had some relevance or importance to Canada. Why 150, you might ask? When we started planning this project, it was last year, which was Canada's 150th birthday. So we thought that was a very fitting number to choose, and in our sense, our gift to the country to sequence these genomes. What else? So yeah, the idea was to sequence 150 new genomes that are relevant to Canada. So hopefully to kickstart some pilot projects for people who can't get access to sequencing, help in understanding the biology of these species, and help with conservation efforts. We have a number of partners that were involved. So CGEn as an entity, we don't fund, we are not funders, we are just enablers. We don't get funded from the federal government for doing research. They're very strict on funding us for operations and infrastructure support. So I had to go out and raise money for the project from various partners, which surprisingly was easy given the narrative of what we wanted to do. So we wanted to keep everything fairly simple. We didn't want to create too much of a burden on people who might want to get their genomes of interest sequenced. Process-wise, it's a very simple 2-page application process that the applicants fill to nominate or to apply to sequence a species. We review those applications on an ongoing basis, on a first-come, first-served basis. Once reviewed and if approved, we have some initial meeting with the project team just to understand what they expect from the project and what will they do with the data, because last thing we want is we sequence, we give them the data, and it sits somewhere under a PI's desk. And then once that is done, the project starts. The key here is that all the data that we produce under the CanSeq 150 program will be globally freely available to anybody who wants to use it. So in a minute, I'm going to show you a few of the things that we have already selected for sequencing. If there's anybody who's interested in getting access to the sequencing data, please get in touch. There's about a 6-month lag between generating data and making it publicly available. That's just to give the people who are interested, who have actually applied to get the sequence done, a little bit of a time to get their head around what the data is, and then it's publicly released. The raw data will be publicly released. So for sequencing approaches, we're looking at it as on a case-by-case basis. Depends on the type of species in question, but we largely put them in 4 bins. We've been talking about bins a lot, so I think I wasn't even planning to use the word bin, but I am. So we put them in 4 groups or bins. The ones that are fairly simple, small genomes in our experience, we use 30x Illumina depth coverage, and 10x Chromium technology to generate the library prep. I'm not going to go into technical details, but again, if you're interested, let me know. Chromium essentially is it replaces the Illumina library prep process, where it hijacks one of the barcodes that Illumina provides, replaces one of its own, and that gives you the ability, in short, the ability to in silico assemble the shorter fragments into one longer fragment. So kind of pseudo long read data. And in our hands, it works fairly well. We have sequenced and published some data on simple de novo genomes on that. Then there's the large genomes that short read Illumina plus 10x genomics is not enough. And in those cases, we tend to generate a little bit, or depending on how much needed, PacBio data for those people as well. Then we have the category what we call the partially complete genomes, and these are genomes that people have worked on before and have some data produced, but they are finding it difficult to complete the genome because there's not enough data. So we help them generate more data. In most cases, it's long read data, because people tend to start with short read data and see what they get, and then they get stuck, and then they want long read data. So that's for those people, mostly we're doing that. And then there's the complex genomes, the trees and some of the fish that are an absolute nightmare to sequence and assemble. And for those, we are trying to get our head around how best to do that. What's the fastest, quickest, cheapest way to do that? And we are putting expertise from all of our 3 nodes together. We are also talking some international partners and see where there's interest then we kind of divvy up the work, saying you do the short read, we do the long read, or the bioinformatics support for that as well. Our three nodes are historically embedded in hospitals and medical institutions. We have a lot of experience with working with human data, but with all other species, we are just, since the existence of CGEn, we're trying to now delve into the other species. So that's kind of the sequencing approach we are taking. So what have we sequenced? Now, please, I am not an expert. The circles don't mean it is these animals, but my staff tells me that this is where I have one person who used to be a taxonomist, that this is where it should be. If you find that some of the examples I give you don't fit where they are, please let me know. Don't shoot me. I'll go and shoot them. So, so what have we done? Just give you an idea. The red circles tell you the species. So we've selected about 51 species by now. So we started in February. So we've already reviewed and approved 51 species, 99 to go. What are we sequencing? Obviously, you cannot sequence de novo species in Canada unless you start with a gray jay, which is now again called Canada jay. Used to be called Canada jay, and then apparently in the 50s, the name was changed to gray jay, and after a lot of effort, we've taken it back, and now it's again, since June this year, it's again called Canada jay. So we're sequencing the Canada jay. The goshawk is our equivalent of the bald eagle, so of course had to be sequenced. Canada lynx and the white lion, I don't need to tell you why. Sitka deer and the wood bison, just for its interest from the conservation and food industry. Dall sheep, stone sheep are up in the northern areas, western northern areas, and very unique to Canada, apparently. The bottlenose whale for its diving ability. So the snowshoe hare and the lynx, you might know, that has a symbiotic relationship. And it's interesting, I got interested, started reading on it. Apparently, the population of the two species go up and down over years together, but the lynx population is trails about 2 years in the sequence with the hare. So I just think because there's not enough food and a lot of them dies and blah, blah, blah. But I don't know. But there's some symbiosis going on there, and they're trying to figure, I say we, but we're doing the sequencing. The PIs are trying to figure that out. The marmots and the woodchuck genomes are also been selected, amongst others. A small toad, again, because of its ability to survive extreme cold temperature. So a lot of these are because of, there's a fly in here as well, the Arctic fox, of course. A lot of these, there's a common theme, the common theme being cold weather, Canada, survival, right? So we're trying to some of it is conservation, some of it is just understanding the biology. Grizzly bear had to be done. The seaweed fly is the one that can survive really, really cold temperatures. Always not freezes to death, it freezes, but then it comes out kind of like a cockroach. Maybe we should do a cockroach too. But it has no relevance to Canada. The weevil and the beetle, a whole list of different fish. So most of these fish are the ones that we call the incomplete genomes. So there's work that has gone on to sequence these genomes, but the assembly of the genomes is not really good or complete. And we were talking yesterday about what does a good or a complete or a near complete genome means. We're still learning that. Again, if you want to talk about human genome, I can tell you all about it. But for other species, we're just kind of getting my head around some of these things. The two sturgeons, some of you might know, really, really old species. They're called the living dinosaurs. They live for 80 to 100 years. Very, very complex genome. I think it's a hexaploid. It's an older genome. This is going to be an absolute nightmare to do anything with. But we're going to give it a try and see what happens. Fortuitously, we had two applications, one for the lake and one for the white sturgeon, from two different groups. So we jumped on it because now there's two species we're doing, but also hopefully we'll be able to use the data against each other to use the data. So I'm not looking forward to the discussion with my bioinformatics team when we come to this one, but we'll see. Snow crab, Canada 150 rose, again, common theme being winter. So this is a rose that survives harsh winters, bred specifically for Canada. It was selected as the rose for the Canada 150th birthday. I have some in my back garden. They're amazing. They're still flowering. Winter comes, they get buried under 3 feet of snow. Spring comes and flower comes again. It's just fantastic. And of course, the sugar maple. Sea star, again, it has a sister species at the Australian coral reefs. And that's why apparently they should be the same, but they are not. There's some difference, genetically they should be the same, but there's difference the way they reproduce. So there's that interest from the PI with their partners in Australia. Sea lions, harbor lions, bats, a couple of bats for conservation, the maritime shrew, the smallest mammal alive apparently, and a whole load of other species. Again, I went through that fairly quickly because I don't want to bore you with all the details, but if you saw something that either you're interested in or would like to know more about, let me know. I'll tell you what I know, and otherwise I'll introduce you to the PI, and I'll tell you where to get the data from once it's publicly available. So again, the applications have come from a wide variety of people. Last slide. Academia, obviously there's a lot of academics who are interested. We're working with zoos and museums, Toronto Zoo, the Vancouver Aquarium. Some of the samples are, as you can imagine, really hard to get by. We got lucky with the dolphin because apparently the dolphin gets us a health check every year. And we just got in at the right time, so when they were taking some blood, we got a little extra few drops of blood from the dolphin because they're in captivity and difficult to get, well, otherwise as well. But profit, non-profit organizations, federal and provincial organizations, oceans and fisheries in Canada and others, and general public. So I even had emails from a couple of school children who heard about the program and wanted to nominate some species, which is absolutely fabulous for the younger generation. Of course, a little bit of challenge because I have to go out and find a researcher, a champion for the species, and ask them to apply for it because somebody's asking for the genome to be sequenced. But it's been very good for that purpose as well. I think that's it. Yep, thank you very much. If you have any questions.